the scientific director um, of the National Institute on Aging, who is also a faculty member in epidemiology and public health, um, is going to speak with us. And I think, as Dana mentioned, following Luigi's talk, there's a group of scholars, we call them junior faculty scholars who are part of the Pepper Center or affiliated with it, are going to meet with Luigi and Jack and Mary Claire Rogman, who leads our research education core. And this actually came about because Mary Claire and Jack invited Luigi to speak with the scholars, and we did a little arm twisting and persuaded them to let us sort of get in on this great opportunity of Luigi speaking. Now, then, after Luigi speaks, those not going in the separate breakout group with the scholars, we're going to sort those that would like to be involved in discussion from multiple disciplinary perspectives, because we have many different disciplines represented here, from those multiple perspectives to get together in small groups and reflect on what they heard from the perspectives of their disciplines, which may be different from some of the others in the group. So it's really a way for people to connect with one another and reflect on some of the issues that are raised. So I'm not going to talk anymore. And we're going to turn this over to Jack Koralnik, who is a faculty member in the Department of Epidemiology and Public Health and a longtime colleague of Luigi's, to introduce Luigi. Jack?
Yeah, thanks, Jay. It's a pleasure to have Luigi here today, and I could spend a long time introducing him, but I will try to keep this brief. Luigi trained in medicine and geriatrics in Florence, Italy. He then showed up at our doorstep at NIA over 30 years ago saying he wanted to also learn epidemiology. And he worked with my group at NIA, an intramural epidemiology research group, for many years, going back and forth from Italy to Bethesda. He then started the In-Chianti study, and he started that study really on a shoestring, very little money, and doing a tremendous amount of work himself. And that study, as many of you know, has grown into a really highly respected longitudinal aging study and is well known throughout the world. His next big transition was when he came to the US for good and to Baltimore, accepted the job as the director of the Baltimore Longitudinal Study on Aging. And this is, as you all know, is a very long-running study, but it was really starting to run out of steam and was not very cutting edge. And Luigi came in and did just amazing innovations and really resuscitated that study. And it now is just a fantastic study with many, many important innovations that Luigi brought to it. His next big step was that he was appointed scientific director of NIA. This is a role where he is basically the leader of all of the intramural labs, including all of the basic science labs. And his extension of his knowledge into basic science and neuroscience has just been amazing to me. He's now fluent in things that most geriatricians don't know anything about. And so if you were to ask me or many people who have worked with Luigi, who is the person with the most knowledge of aging across a very broad spectrum of subjects, from basic science through population science, epidemiology, all the way up through clinical medicine, clinical geriatrics, I would say definitely Luigi, and many, many people who he's worked with who have been had the good fortune to work with him would say the same thing. So we're fortunate to have him here with us today. And Luigi, I know it's 10 o'clock in Italy, but hopefully you can keep your energy up for the talk. And we thank you for joining us.
Thank you, Jack. I had some good Italian coffee, so hopefully I will not have a problem with that. Let me start by showing some slide, and while I'm doing it, thank you for this really nice invitation. So I want to start with this slide that is something that I learned a few years ago. And I learned that in Japan, broken objects are often repaired with gold. And they think that the flow, the broken and repair, is seen as making the piece unique. And it's kind of tracking the object history with art to its beauty. I think that that to me is an allegory to what's happening to aging. We go through our life through an infinite number of damage and repair. And this damage and repair make us unique in spite of, for example, identity of genomic material that identify twins. So when they got to the 80 or 90, they're as different as people that have not any genetic identity. And so aging, in a way, is what makes us who we are. Of course, with some side effect. They may not be always pleasant, but also many, many aspects that are really interesting and peculiar to study. So I want to start by looking at how the concept of aging has changed in the last few centuries. And I want to do this by showing you two paintings. The one is from George Latour that show the old man. We are in the 1600s. As you can see, a man that is stooped with the support, with somewhat swelling legs, looking around a little bit disoriented, with a white beard, no hair, and really showing the sign of old age. And then we go much more recently to the beginning of this century, 1932 or 34, with Kazimir Malevich that is drawing the running man. The running man is old, but his white hair and white beard are flowing in the wind. He's escaping from the cemetery, he's escaping from death, and is showing, incredible joy and incredible energy. So our concept of being old has really changed dramatically, coincidentally to the change in our physiology and in our life expectancy. And this transformation, to me, it's very well explained in this beautiful slide that I took from the website of the United Nations. And I just want to show you how, for example, this blue sagoma here is the structure of the population. You see that the ears are on the left side, and here you have, a tracking of time. You can see that the people over the age of 70 in 1950 really were a very, very small number, very, very, tip of this triangle. And as you grow older, from 1960, 70, 80, 90, and 2010, and then projected to 50 to 75 and 2100, when we go into, the yellow part, both for men and for women, you create big shoulders. And these big shoulders represent, large number of older people that are populating the world, and not only the developed country, but rapidly and very, very fast, and somewhat unexpectedly fast, also the developing countries. Another way to look at the aging of the population, it's this slide that comes from a work from Jean Vaupel. I don't know if you know Jean Vaupel, but Jean was one of the, probably most famous and most notorious demographer studied aging and recently died. If I'm not wrong, he's actually died in the March of this year. And he was probably the greatest demographer that studied aging over the last 50 years. And what he and this group did was to plot, the year, the epoch here that you see on the x-axis, and on the y-axis, the life expectancy here. And here are the cities and the country with the longest life expectancy at that time. And as you can see, these are, on a very, very incredibly straight line. And over the year, people that were looking at this straight line, trying to make a projection, have you lost me?
Yes, we did.
When?
Talking about the straight line, making a projection from the straight line.
Okay, so people were saying that this straight line could not be continued. And so the UN started making projections that are illustrated in these red lines. And as you can see, the many projections that people made in different epochs were not validated. And the oldest population kept going on and on and on a straight line. I don't have an update slide, but in 2022, we're still on a straight line. So if we look at different population over time, the aging of the population doesn't seem to slow down or to stop over the last few years. So this is incredible success. The aging of the population is something that we need to celebrate. But of course, as I said before, come through many twists, some positive and negative things. For example, one of the things that we are realizing is that the aging of the population is not homogeneously occurring in different parts of the world. Here I focus on the United States. And for example, you see that around the 60s, if you look at the life expectancy of birth in the different states in the US, the range of life expectancy was around, in the range of plus or minus 5.6 years. And you can see that over the year, although the life expectancy is improving, is increasing, the range is also expanding. So there is a problem of health disparity that is occurring at the state level. And here I show you two examples. One is Connecticut and one is Oklahoma. You can see that from 1990, the increase in life expectancy in the state of Oklahoma was negligible, while in the state of Connecticut, you see that the increment in life expectancy was almost linear, suggesting that, yes, there is improvement in medical care, but there are also important environmental conditions that affect how long we live. And of course, it is important that we try to understand how we got here, to understand what's going to happen in the future and how to address that. This is a very simplified slide, just for me to introduce what I want to say. So think about the pre-industrial state. I don't put any date here because different countries have gone through this phase in different years. And in this pre-industrial steady state, you have the high fertility and high mortality. So fertility and mortality were very, very similar. And so they were balancing each other. However, with the improvement in the living condition, and probably the most important factor was the availability of water and clean water and food for everybody, you see a substantial decline in mortality. And at least initially, the decline in mortality was due to, infant mortality decline, infant or small or young people mortality decline, while the fertility remained high. And the fertility remained high because the industry, still the farm industry, where having many children is still incredibly useful and important. At some point, there is the phenomenon of industrialization and especially urbanization, where you don't need those children. And so you see the fertility decline, and the fertility decline really, really fast, and declines so fast that at some point we have seen in many countries, or we expect to occur in many countries, a crossover. That's something that not many people realize, though those that are not interested in aging, but think about that if the fertility decline below the mortality, that means that the population is shrinking and will be further shrink in the future. And how much shrinking we're going to see? This is, again, from the World Population Dynamic in 2019, and this in the Ukraine war era. You see that many of the East countries, Bulgaria, Lithuania, Latvia, are projected to have a decline in the population by 2050 of 20%. 1/5 of the population will disappear, and those who remain will have a very, very high percentage, old people. You can imagine, in spite of the increase in health, what will this mean with the economy and the change in society in those populations? Another problem is that the decline in mortality that we have seen is mostly due to decline in heart disease, stroke, and to some extent also cancer. I think that the acceleration in the decline of mortality by cancer have been seen in the last few years with some of the success of the immunotherapy. And I think that we will see a decline in the future that is even stronger than that. But what is absolutely not changing, what our society and the medical field has not really been able to address are the older people with complex multimorbidity and disability. If you look at the mortality by influenza, pneumonia, Alzheimer's disease, chronic respiratory disease, depression, diabetes, complication of diabetes, not only they're not declining, but they're slowly and progressively increasing and are projected to become major cause of death as we go toward the end of this century. And if you look at the number of people that of a non-nursing home population, somebody has something? Are you okay? Can you hear me? Okay. The percent of non-nursing home population age 65 and over with dementia, you see that, it's really changed dramatically. Between 2011 2015, there's already an increase in 2%. These are just 4 years. You can see an increase in 2%, especially in the men. And so I think that you can imagine how this is going to affect, the composition of our society. And finally, and then I'll stop with the, illustrating general data, but I think this introduction is important for me to introduce what I want to say later. This is a slide from Eileen Crimmins, really great demographer and epidemiologist, showing that the change in life expectancy between 1998 and 2006 was illustrated in this green bar, but the change in the percent of life expectancy that people live with chronic disease has increased much more, which means that we have seen an increase in longevity, but all the longevity or even more is actually attributable to the life that people live with chronic disease and disability. So there's something that we need to do about that, and something that we need to do to frame and target that population that end up being with complex multimorbidity and disability. So I'll start telling you what I think should happen and what we know about what's doable by showing you this slide. And this slide comes from somebody that in one of the most recent conference criticized me because I always show, all the people that are declining and become stooped and cannot work anymore. And they wanted to tell me that there are older people that are incredibly successful with multiple perspectives. And you will recognize some of these people. I love this picture, and I want to challenge you to tell me who this person is at the end of our conversation. But this slide show you how even if you select the most successful people, the effect of aging is pervasive and incredibly powerful. Now, we have always thought that we need to study aging by studying this part of this population, the one that has this, yellow frame. And I think that that's right. We have done that, under the guide of Jack, on many studies looking at disability and mortality and, the relationship between disease and disability in this population. And we ended up, I would say, in a nutshell, to realize that in older people, we can really characterize their health pretty well by doing what? By measuring disability. And for example, this is, probably the most cited of my paper that is from Jack, showing that, the very test, the SPPD, a very simple test of or lower extremity function in the EPI study was predictive of mobility disability and ADL disability. And I want to point out that this test can be done in 10 minutes, costs very little money. It's immediately interpretable. You don't need to wait until the lab sends you the result of the test. And you can immediately interpret that. And it's incredibly powerful, much more powerful than any blood test that you can do. You see, these people that have a SPPB of 4 have 50% probability to develop ADL disability, while those people with a 12 have less than 4% probability. I mean, that is so direct, it's so powerful that I think that I don't know. When I saw this data that Jack presented me, I was incredibly surprised. And I think over the year, I still present this slide probably every time I give a talk. And then this is a study that we did in the Chianti study. We never published this data because I thought that people would not believe it. But we looked at walking speed at baseline. And then we divided the baseline walking speed into quintiles, and the lowest quintile was the walking at a speed lower than 0.8 meters per second. And then you look at the mortality in this population. We compare the mortality in this population with the mortality of people that at baseline at the diagnosis of malignant cancer. And you can see that this is walking at less than 0.8 meters per second. It's more powerful than having a diagnosis of malignant cancer in predicting mortality. So in that population, I think that at least we know how to characterize health. I'm not sure that we know what to do to improve health yet, but at least we know how to characterize health. This is a study from Stephanie Studesky showing basically the same thing in multiple populations, that even a 0.1 difference in walking speed is associated with higher mortality. And then I want to go back to a study that we did with Jack when we were working really close together. And I was constantly going to his office to get help in the interpretation of the data. So this is a study that was published in the Journal of Gerontology and Medical Science in 1996, where we wanted to see, at that time, at least for me, when I was thinking about somebody becoming disabled, the image I had in my mind was somebody getting a stroke or somebody getting a hip fracture. And this was biased by the fact that my experience was working in an intensive care unit. So I only looked at those kind of disability. But when we look at that and we look at whether people were becoming disabled progressively over time or catastrophically, in a very short period of time, we found that a substantial percentage of people are actually showing a long and progressive trajectory that lead them to disability. And this percentage of progressive disability was much higher among those that were older, 85 and older especially, but you see some sort of a progression even in earlier age. And then we look at what happening to them during that period that they become disabled, and we created a window of time that was similar for the progressive and catastrophic disability, and we look at hospitalization that had occurred during that time. And you can see that in those that were showing catastrophic disability, there was a very large number of diagnoses. 50% or more really show a potential cause. Well, in those that were showing progressive disability, 25%, 27% show some potential cause, but even those causes were not really the cause I was imagining, not only the fracture, but there was dehydration, diabetes, pneumonia, multiple, and a large percentage then had no hospitalization or had a very large variety of causes. None of them configure the idea of catastrophic disability that I had in mind. So that means that really to understand this progressive disability that becomes such an important cause of disability in old age, we need to think longitudinality in terms of trajectories. And so I think that one of the secrets that I understand at that time was that, and more later on, was that we also need to look at what's happening here. I think that the history of what's happening in old age really lay on the trajectory, in a life trajectory. And we know now that you need to go earlier during, the early years of life, or even during the gestation. Okay. So what's happening? What's happening that during the lifetime make people disabled? If we want to maintain our attention to the clinical side, one of the things happening is that the people tend to accumulate disease. And one thing that we realize, and this, I don't have the slide from the age of 20, but you will see that this is working even from the age of 20, you will see that the number of chronic disease over time increase with aging. But when you look at longitudinal cohort, in the accumulation of multimorbidity, you see that the trajectory becomes steeper and steeper. And so that the accumulation occur not linearly with age, accelerate with age. And people that develop one disease that then tend to develop another disease and so on, suggesting that there is a global susceptibility to disease that occur with aging that we have not really considered. And that's really what makes complicated to study and to understand older people. And I have for you, a fantasy slide. This is not real data. So somebody is what's going on? Jack, do you have a question?
Somebody was unmuted.
Okay. So imagine one person. This is one person on top. That is the average person. You know, this person will develop pathology for osteoarthritis, and then at some point, that pathology will cross the threshold that will make them detectable at the clinical level. However, you know, we used to say that what will happen if we eliminate cardiovascular disease? What will happen if we eliminate osteoarthritis? And in reality, not as much as you can imagine, because what we're finding is that disease tend to develop in clusters. So the people that have one disease have much higher probability to develop a second disease, and two diseases have a much higher probability of developing the third disease. And so that even if we push the first disease, as we suggest here with the CHD that come later, I have pushed a little bit, but in reality, the cluster manifests itself. And what I really want to do is to reach a condition where this global susceptibility to disease that is due to age is somewhat addressed. So there is and this is the shift that has occurred in the field. Is the idea that I will call the geroscience idea, that in fact, diseases are not else than the clinical manifestation of aging. And the people that develop multiple disease early in life, on average, means that they have accelerated aging. And the people that are late in developing disease are the people that slow down aging. This is, to me, the best, vignette that from Seals and Mello, saying that, yes, we can do specific prevention of disease, but if disease occurs in cluster, we just have to wait from the next disease to manifest. Or we can try to act on the root and slow down aging. And by slowing down aging, we will develop a strategy that prevent and delay the many chronic diseases to occur. But really the problem is that, can we slow down aging? I remember talking with Andrea Lacroix, when I came to NIA, and she told me, well, you really need to adjust for age because age and sex are immutable. And so the only thing you can do is account for them. Well, we do know now that that is not completely true, at least in model organisms. We can actually modulate aging. And I don't want to go into the animal model, but I want to illustrate what is the conceptualization of aging that allow us then to try to find the tool clinically to demonstrate that aging can be modulated. This is the framework that I always use when I think about or plan a new study. And I will going to go through this quickly because otherwise I can spend a lot of time in it. But imagine that there are two opposed forces. One is the entropy that creates damage, and the other is resiliency. And the resiliency opposes the damage and repair what has been damaged. And when you are born, you have a lot of resiliency. And so the aging is not even visible because the program implemented by development is so strong that you can't even see aging. Aging is occurring, and mostly because the stochastic entropy is also affecting the mechanism of resiliency. And at some point, you get frayed in the sense that even the mental stress of life is not doable, is not feasible when you are in the frail condition. You can have accumulated aging that is faster than usual through two mechanisms. And the first mechanism is by, being exposed to a lot of damage, to a lot of stresses, for example, radiation, or infectious disease. And so you hit your resiliency faster than you expect. But the other way could be that you lose resiliency faster than expected. And if you lose resiliency faster than expected, the effect on early mortality and frailty will be the same. One of the issues is that we don't know how to measure resiliency yet. We know how to measure damage because this has been the focus of traditional medicine, but we don't know how to measure resilience over time. In order to measure resiliency over time, we need to take a longitudinal perspective. And I want to stop a minute on that because I think that it's basic epidemiology 1.0, but it's still worthwhile looking at it and remembering. So usually, many times, in order to understand aging, people are comparing groups that are of different age. So people in their 20, 50, and 80. And not on shock, when they created the LSA, they say, well, this is not really right. What we need to know is to follow people longitudinally over time, because that's the only way to really understand aging. And I wanted to show you a few slides that demonstrate that this is actually the case. So the first slide is this one. So imagine that we are comparing 4 individuals, 20, 50, 90, and 105. And we think that those 4 individuals are different. And so we say, well, this is an effect of age. However, this individual at age 105 was 20 during World War I. And you can imagine the amount of stress that people in World War I were adjusted to. This person in the age of 90 was during the Great Depression at the age of 20. They didn't really have enough to eat. They didn't have clean water. And why this person at the age of 50 was born during the Green Revolution, which was the first time that theoretically there was enough food in the United States for everybody to avoid to be angry. It did not happen, but there are social constraints that I will not discuss here. Why this person at the age of 20, at the time when I created this slide, was in 2000. I actually created this in 2010. In 200, there was the lowest rate on unemployment. There was around 3.8%. So these people are not only of different age, but their experience of life is so incredibly different that we can't really claim that the only different is the age. And so there are secular trends that truly affect your comparison. The second issue is selective mortality and selective participation. So this is a real data. There's a number of chronic conditions in people at the time of entry in the LSA. You can see that the number of chronic diseases increased with aging. However, when you look at their trajectory, longitudinal trajectory, the longitudinal trajectory are quite different. And the accumulation of multimorbidity is much steeper and faster than expected based on the cross-sectional projection. This may lead to a number of mistakes and a number of misinterpretation of error. This is a very famous study, not mine, was done in the LSA by Jerry Flagg and Ed Lacatta, showing that if you look at VO2 peaks, the maximum fitness longitudinally and cross-sectionally, you see that the decline with aging is much, much faster than expected from cross-sectional data. And this is because those who remain in the population or they participate in the study are those that are healthier and they're not the sick people. So from your analysis, you eliminate the sicker people in the population. This is another study that we did a few years ago, looking at white blood cells. And if you look at the white blood cell by age, yeah, there is a stratification by age. But you see that there are very, very important secular trends that confound this estimates. And let me go directly here. And even though there are differences, the effect of white blood cell on mortality is maintained, the order, the rank is maintained, but you can see that the absolute values of mortality rate are substantially different by different years. So we can't really look at cross-sectional data and pretend that they are cross-sectional. So what we need to start looking at is what are the changes that occur with aging, and how, by looking at this changing, we can predict how people slowly and progressively develop some disability. And because I don't have 10 hours, I'm going to look at and show you only a few of them that to me are particularly interested and have been important in my study. And the first thing I want to talk to you is resting metabolic rate, or basic metabolic rate, which is well known for many, many years. This is a publication on 1919. So the resting metabolic rate has a very important trajectory with life. So it increased tremendously, in the first couple of years of life. And you can imagine why, and increased tremendously because you have to build protein. And in building protein, you need a lot of energy to construct your body. And then tend to go down. There's puberty, and during puberty tend to go up again, and then decline, and less slowly and progressively decline with aging. This slow and progressive decline is something that I was reading when I became the director of the BLSA. And I say, well, if this, resting metabolic rate decline with aging, which will mean that those that maintain high resting metabolic rate will do better than those that do not show a decline in resting metabolic rate. This was my theory. And of course, I was completely wrong, and the association was absolutely the opposite. And we found that, in fact, high resting metabolic rate was a risk factor for mortality. This is important because that shows you that what is happening over life is that you use part of your energy to fight the deterioration of the soma that occur with aging. And for example, here you see that there is a dissociation in the rate of resting metabolic rate in the BLSA from those that are not healthy and healthy. I can go on and tell you how we define healthy here, but it's based on the definition that is the entry criteria for the Baltimore longitudinal study that we call the ideal criteria. So how do we interpret this data? Hello? I can't hear what you said. Hello?
Luigi, can you hear us?
Yes, I can hear
Okay.
you. Can you hear me?
Yes, fine. There was just
There
some
was
extra
something, I
noise.
don't. Okay, just noise. Okay, so how do we interpret that? And this is a complex slide, but I want to make it pretty simple. I think that imagine that during the day, you have a package of energy to use from morning to evening. And then you receive this package in the morning, and you have to use that for the different activities. And 60% of it you use for stay alive. Okay, that is as much as you need just to stay alive. Then you need some for maintaining autonomy, dressing in the morning, going to bed, eating something. And then you have the little bit that is there for you to have fun, reciting poetry or dancing or doing whatever you like to do. Now, with age, there are many things happening, but what I want to focus here is that there is a quota of extra energy that you use to maintain health. So the more challenge your body is, the more pathology you're accumulating, the more you need energy to maintain homeostasis. And there are many demonstrations of that. One of that is that, for example, to walk 100 meters at the age of 80, you need more energy than at the age of 20, because your efficiency of your mechanism, your mechanics of gait is altered. And so that means that your resting metabolic rate increase means that you are coping with pathology. And that is why higher resting metabolic rate over a certain threshold is associated with higher mortality. And so it's a behavioral compensation that is associated with mortality. Okay. Now, you think that this resting metabolic rate may not be so important, but look at, oh, this is going by itself. I'm sorry. These are, since data was from the Baltimore Longitudinal Study of Aging, showing that VO2 max, which we may imagine is the maximum energy you have, decline with aging. And then we say that the resting metabolic rate increase, and then means that at some point in life they converge. So you need to use most of the energy you have to stay alive. You don't have any extra energy to do anything else. And because of that, you start developing this progressive disability. So at least in part, the progressive disability is a progressive discrepancy between energy availability and the energy demand in your body. And this is so true that this is a very, it's not very, it's an old slide that, 18 years ago. And this shows you something very clear. So here you have the VO2 max and normalize the 100% VO2 max. And so at preferred walking speed, at the age of 80, your preferred walking speed is already eating more than 60% of your VO2 max. Whether at the age of 65, not that much younger, is less than 40%. So as you go to old age, these black part get closer and closer and closer to the VO2 max, which means that you barely at some point only the energy necessary to stay alive. And that is the condition of frailty. And that, I think, that's my interpretation, is why exercise is so powerful, because exercise expand the VO2 max, expand the amount of energy you have. And so take the threshold from the, preferred walking speed to the VO2 max, expanded. I'm gonna skip this and this too, because I see that we're getting late. I wanna tell you something that is somewhat peculiar, and that we need to start thinking of the complexity of capacity and behavior. So this is a paper that we published with Teresa Tian very recently, looking at walking speed as a predictor of dementia, okay? And we looked at two parameters of mobility. One is walking speed, and the other one is activity fragmentation. So what is activity fragmentation? It's a complex formula, but the concept is extremely simple. An older person have to do everything to maintain autonomy. But because of the low threshold to fatigability, they may do it in small fragments. They may do something and then rest, and do something and then rest. And the more they fragment their activity, the more they are able to do and to maintain autonomy. Now, the fragmentation, it's a conscious decision. So what we found is that walking speed was associated with dementia only in people with low activity fragmentation, not in those with high activity fragmentation. And we were really puzzled by it. We didn't really understand what was going on until we started thinking that fragmentation is a conscious volitional compensatory activity. And so if you think about low walking speed is a functional measure that is due to many different things. Some of them may be caused by dysfunction in the central nervous system, and some of them may be more biomechanical. For example, pain, osteoarthritis, or sarcopenia, or fatigue. And so you expect that the people that have a problem in the brain are much more likely to develop dementia because they already have a problem in the brain, but are much less likely to fragment because they cannot use that conscious activity. And so the lower fragmentation is suggesting that they're unable to implement this compensation. And because of that, they show that there is a cause that is more neurological than structural, and that associated with dementia. I'll walk you through again in this paradigm because it is, and we're currently doing study to demonstrate this more formally, using VLSA data. So let's say you have full mobility, and then you have mobility impairment that is physiologically compensated. You, for example, enlarge the base of support or fragment, and then you behaviorally compensate by fragmenting your activity. And then at some point you cannot really do that, you develop mobility disability and then ADL disability. So you may have two situations. One is that fragmentation you are compensating reveal that you already have a problem. It may not be seen by walking speed because you're compensating, but when you look at the compensation, it reveals to you that you're having a problem. However, if you cannot compensate, because, for example, you've lost behavioral plasticity, for example, for reduction of brain function, that indicates you have a brain problem. And this brain problem eventually will be coming out as brain pathology and cognitive decline and dementia. So you see that how interpreting different type of mobility parameter from modern technology, we can implement more and understand more the nuances of a test as simple as walking speed as we can think of. Walking on mobility has been, most of what I've done with Jack, and I'm still very much curious about how we can exploit mobility. This is just an historical perspective. This is a Sunday morning, Baltimore, Maryland, in June 6, 1948. This is not June 6, but I think we're very, very close. You can see that there's a US population predicted to change in proportion of various age group from 1950 to 1980. But what is peculiar to me is that the vignette that represent this change is really looking at people that are walking or looking at their mobility. So not very much it changed from that. And then the last word, see, the problem with doing this to prolong your life is that actually you come at the end when you're old. Thank you for your, for following the presentation. And I didn't put any of the biological part, but underneath that decline that was showing you before, we now know there is a complex network of biological mechanisms that we're starting to scratch on the surface, and eventually will provide target to prevention, to the promotion of healthy aging.
Thank you, Luigi. Thank you very much. Um, Why don't we see if there's a couple of questions or comments and then decide what we want to do with the breakouts, given the time
So sorry, it took me a little bit more, but we started late also.
it's okay. You don't have to apologize, Luigi. It's all good information. And it didn't seem like a long talk to me. It was very.
I would love to ask a question, Dr. Ferrucci.
Go ahead.
Hi,
Hi there, Chris Ward. That was just a fantastic talk. I'm really interested in that intersection of the drop in VO2 max. The idea that exercise can increase, VO2 max. If you placed on that relationship muscle mass as we age,
yes.
would that track with the?
That's a very good question. I can tell you that 50% of that decline is due to the change in body composition, but it's then another 50% that we do not understand and we can't really adjust for. So there is a decline that is not due to muscle mass, or even when you put in it muscle quality, it doesn't go away.
But when you then exercise, is the improvement mostly predicted by muscle energetics?
Yes.
So that's the improvable part. The other part is kind of still a mystery.
Yes. Yes.
Yeah.
Absolutely. Absolutely.
Yeah, that's fascinating. Fascinating.
Any other questions for Dr. Ferrucci?
So I want to say one thing to Chris. Remember that when you exercise, you are not only increasing the energetics of your muscle.
Yes.
Very much you're increasing energetic everywhere in your body. I think that people are starting to do P31 spectroscopy in the brain.
Yep.
When people exercise, the P31 spectroscopy in the brain improve even in the fat and even in the brain. So I think that's something that we not completely understand yet, but I think it's part of what's missing.
I would like to ask a question if that's okay.
Alonzo.
Yeah, thank you, Dr. Ferrucci. It was really nice, right? A lot of interesting information. And I'm gonna just ask like a really broad question, but just curious because you show like a lot of factors that may influence age. And I was just curious in your experience and your point of view, which physiological, biological factors do you believe are the more important ones in your experience? I know it's a very broad question. I'm just curious what you would like name the ones that you think really impact aging.
So, first of all, I think that for me, aging is a problem of energetics. That's my belief, but I can't really give you a formal, complete demonstration. I think that what's happening is that the more there is, so think about when I talk about resilience, okay, and bring that resilience to a molecular level and to a cellular level, which means that you have a DNA damage, you repair it because there is a beautiful network of molecules that repair the DNA. You have a misfolded protein, you refold it. You have a mitochondria that is broken, you eliminate that and you do mitochondrial biogenesis. All of this is happening to your body. But what that need is energy. You cannot do resilience, you cannot repair. You don't have enough energy to do that. And that kind of energy can only come from aerobic metabolism because the glycolysis is really so little efficient that we'll never be able to pull that energy. So to me, if you think about what is the part of the body that is driving the system, it's energy, is the energetics. That's why lately, what I really study a lot is mitochondria.
Okay, thank you. Thank you.
Any other questions for Dr. Ferrucci?
Hi, this is Les Katzel. Do you want to please comment on the impact of selective mortality on the multimorbidity and disability, particularly in the oldest old?
Can you tell me again? I didn't
The,
understand
the,
the
the
word.
impact of selective mortality with the people who have the multi conditions are dying off. So if anything, you're understating their impact.
Yes, I was trying to suggest that, I think that unfortunately, we have the very few population that have been able to follow very, very old people up to late life. There is some emerging data from the UK Biobank. There is data from the registry in the Nordic European countries. And we know that by looking at that, our estimate of multimorbidity and mortality associated with multimorbidity and disability for the oldest old are really not working very well. And the other phenomenon is, so I think when you reach that age, above the age of 90 or 95, I think that there is certainly some genetic predisposition that make people different. I'll always this disability and multimorbidity are no longer a factor for mortality at the age of 100. I think inflammation is, but disability is not. Hearing loss is, but dementia is not. So I think that you're dealing with a body where the survival probability is affected by a peculiar equilibrium that may not represent what is maintaining homeostasis in the global life. If you talk to Claudio Franceschi, he will completely disagree with me, but that is the fact.
Okay, thank you. I think we're going to cut this off. And I've been communicating with Ann Sullins, who has been helping us organize today's event. And what I think we're going to do is, given the hour that we've gotten to, I think breaking up into small groups, other than the group with the scholars, the Pepper Center group meeting with Jack and Mary Claire and Luigi, I think others we're not going to have time for the additional discussions. And I'm seeing Amanda nodding, one of our moderators, and Anne and Alice. I can't see Alice, but I think given the hour, we're going to not have those discussions. If anybody really wants to stay on and talk more, we can do that. But why don't we just break and let Luigi meet with the Pepper Center scholars, and also thank everyone for joining us, and hope that they found today's talk quite interesting. I know I did, Luigi. I always learn something new and another way of thinking about it from you. So thank you very much.
Thank you. Thank you, Jay. It's always a pleasure.
Bye-bye.
So is somebody putting me in a group now? I think that
I think everyone will just stay here.
okay.
Oh, and we have a small but select group.
Good.
Thank you, Luigi. My name is Mary Claire Rogman. I am sort of the odd person out in that I'm an infectious disease physician who has a strong interest in education and training. And I do some work in nursing homes, which is how I got brought into our Pepper Center. But I also am really interested in preventing diabetic foot ulcers because of their association with infections. And I've always been very frustrated at watching how downhill my patients will go after developing a foot ulcer. And I wondered if you could talk about how that paradigm might fit with what you just discussed. I can only imagine that having a chronic wound would be an enormous stress on a person.
Well, I can't really talk to you by experience, but I think that so there is a school of thought that your ability to be resilient, for example, through the immune system, is not infinite. There is an exhaustion point. And for example, there is a work from Eileen Kreenings and Tuck Finch showing that in people that are exposed to multiple infection in their life, there is an accelerated immunosenescence. They work with the Aborigine, people that work, that live in the forest, that are exposed to many, many more infection than we are. And so they selected multiple population for comparison, and they found that many parameters of immunosenescence, both of chronic inflammation, but also response to vaccination, are much, much lower in these individuals. So you can imagine that the wound that to stay open would require constant and continuous, response to inflammation and infection. And one thing that we tend to forget, but we shouldn't, is that wound healing is done by the immune system.
Yeah.
It's not that the immune system, also repair. The macrophage from M1 become macrophage M2, and they, deposit, the intercellular matrix. And so they do a lot of work during wound healing. And so we put the immune system under a lot of stress. And I think that the immune system, it's really, people talk about the hallmarks of aging. And the different hallmarks of aging, if you think about when signally, each one of them generate inflammation. But I think that the most important one is the connection with the mitochondria. We just wrote a review paper showing that the place where energy is generated is the hub of the inflammatory response. And the inflammatory response during wound healing is specifically from the inflammasome, because you see IL-1 and IL-18. And so that also means that there is mitochondrial stress ongoing there. And that's why I think that wound healing, wounds that do not heal are truly, factor that can affect the health of an individual tremendously.
Thank you. Other questions or kind of ways in which what you heard applies to what you're doing? You have the opportunity to introduce yourself.
I'm happy to go first, Dr. Rugman. Dr. Ferrucci, I'm Jason Falvey. I'm a physical therapist and work here with the Pepper Center, and I'm faculty in the Department of Physical Therapy and Rehab Science here at the University of Maryland. So as a therapist, of course, I'm thinking, man, some of this sounds really inevitable. Once you develop some of this disability, you're in big trouble and your course seems set. And I always wonder, how reversible is some of this stuff? What do we do well as medical professionals doing restorative care? And maybe what do you think in terms of targets should be really focused on as therapists? We focus on range of motion and muscle strength, but should we be measuring something else?
Well, I'm not really sure I can truly address your question. I think we're still in the exploratory phase where we measure the measurable as much as we can to try to understand what is relevant and what is not. I don't think that we have a complete understanding of what are the most important variables. I will say two things. Among the many strategies that people have used in middle age to slow down some of the effect of aging on muscle, for example, I think that recently one is becoming the only one that seems to work, which is the urolithin A. And urolithin A is has an effect on mitochondria. So that people have been taking urolithin A, and the way urolithin A works, it's really interesting because it does not target mitochondria, but target the mechanism that remove the mitochondria that are damaged so that the organisms created new ones. And so from those people, you see that there is an improvement in muscle mass and muscle strength and muscle quality in spite of no difference in physical activity. I don't think that there is any other intervention except for exercise that have been able to do that over a long period of time. Now, this is just muscle. And you know, problem with the muscle is that it's important, but it's also the one that we can measure. You know, in the other place, we can't really take measure. You know, I will, one of the dream I have, which at some point I hope I will be able to realize, is to measure energy in the brain. You know, is trying to understand and do P31 spectroscopy in the brain and see that whether the intervention and improve the muscle also have an effect on the brain. And these are the two most important tissues for me for many reasons, but the most important one is that they, in terms of weight specific energy consumption, are the two, you know, most important and most valuable. You know, the amount of energy required from the muscle increase 100 times during contraction. At rest, between rest and contraction, the amount of ATP requires 100 times higher. In the brain is the same. You sleep and then you have intense cognitive activity. The consumption can increase 30, 40, 50 times. And so I think that those are the two tissues that for me are the most interesting. But, you know, what do I know? I don't know.
Hi, Dr. Ferrucci. Thank you so much. I'm Jennifer Albrecht. I'm an epidemiologist here in the Department of Epidemiology and Public Health. I study primarily traumatic brain injury among older adults. And I was really, I really loved your talk. I've heard you speak multiple times before, and I always enjoy everything you have to say and talk about. I thought it was really interesting looking at those trajectories of comorbidities that you showed. It was like someplace in the middle of your lecture. Because I've thought a lot about traumatic brain injury as not so much an outcome as much as a sort of just one more accumulation, right? On this sort of pathway that people are traveling, where they start accumulating these comorbidities, and then they have mobility disruption, and then TBI could just be sort of one more thing that happens that might guide them ultimately towards dementia and additional disability. And I was wondering what your thoughts were on that.
So I think that there are two emerging theories on why we age. One is the damage accumulation, which I think that the way I think, but there's another one that is equally interesting, that is the idea that we don't repair because the signaling network that coordinate the complex series of events that lead to resilience and repair is dysfunctional. So it's an information processing. And I think that if you think about the, brain injury, if there is one thing that may affect the information processing, is certainly that, especially if you think that now we know that many of the peripheral activities are driven by a handful of cells in the hypothalamus. There is a little bit more than 1,000 cells in the hypothalamus that control your stomach, your intestine, your bladder, your muscle, the bone, your bone. All those things are, affected by a handful of cells in the hypothalamus. And so, imagine having a brain injury and destroy a few of those 1,000 cells. They're not millions. a very, very small number. And so I think that that's a great possibility. I'll tell you that if I were going back, and I will do the same thing I've done, but if I had to counsel somebody today that start I will work on the hippocampus. I will work on, the deep part of the brain that connect the brain to the other parts of the body, because I think that that's a part that is not being studied now. So I agree with you. When you damage the brain and coordinate all this activity, think about the clock. The hypothalamus clock that synchronize all the other clock in the other tissues. If you dysregulate the clock in the hypothalamus, the clock in the muscle, the clock in the fat, the clocks in the intestine, the clock in the heart are not synchronized anymore. So your body is no longer synchronized. And of course, they start accumulating multimorbidity. But if somebody tested this hypothesis, because it's a testable hypothesis, if we looked at whether the event of brain trauma is associated with increased accumulation rate of multimorbidity, I think this is a testable hypothesis. It can be done in a study.
Tell that to NIA and maybe they'll fund one of mine. Thank you.
Could you do it in animals? I'm sorry, I'm not going to get us off.
Oh, yeah.
Anybody else? Anybody else?
It could be done in animals. I think that I have somebody very, very much interested in that. I should tell him. Well, in animal, except that, how do you diagnose disease in animal? You can, but it's not the same thing that you do in humans. Yeah, people pretend that they can, but I'm really not sure. And I only do study in people, so I don't study anymore on principle.
Janine or Marcel?
Yes. Hi, Dr. Ferrucci. My name's Janine Ursity, and I'm in the Department of Orthopedics, and I work with Chris Ward. And thank you very much for your talk today. Really enjoyed that presentation. And what our work is in the Ward Lab primarily studies muscle. And the project that I am working on in association with the Pepper Center is looking at peripheral blood mononuclear cells as biomarker for osteosarcopenia. And so we're looking at peripheral blood mononuclear cells, looking at the cytoskeleton and looking at cell mechanics to see, since aging has so much of what's involved with aging are systemic factors, we thought that perhaps being able to assay an easily accessible cell like a peripheral blood mononuclear cell, that it might give us an insight into the health of muscle and bone. And so in your talk today, I was struck by, as you're talking about delaying aging. And so we're working in preclinical models right now. We hope, and we actually had discussions with someone at the Wake Forest Pepper Center today about obtaining human samples. And so we're hoping that this work will translate to human samples. But at this point, working with animals, we've accessed animals from the NIA colony, and we've accessed them from the Jackson Laboratory colony, and they're very different. And so what we see is that the NIA colony seems to have delayed aging because if we get animals from either facility at the same age, whereas the Jackson Laboratory older animals might look very old and have a lot of features of, sarcopenia and, other, and we're able to recognize some changes in the PBMCs. The NIA animals seem to be quite healthy at those same ages and not express those same factors. And I was just wondering if we have some ideas about what might be going on, but didn't know if you might have any comments or advice for us as we work through the preclinical stages, accessing the different aging colonies.
Yeah. I mean, you asked me something that I'm certainly not an expert on. I'm not an expert in animal model. I can only tell you what I see and what we discuss with the other group within NIA. One thing that we have realized working, especially with Rafael DiCabo, is that the idea that because two animals are genetically identical, reduce their variance in their phenotype is an illusion. In fact, if you take a bunch of mice that all the same genetic background, and then look at the variability of some of the phenotype, weight or body composition, we see exactly the same variability that we see across different species. So because the epigenetic, it's so fundamentally important to really affect the phenotype. And because of that, then you start thinking what is affecting the genetic, the epigenetic, and the epigenetic is affected by the environment. It's what these mice eat, how much light they get, how much they are in a cage, two together, three together, four together, how much cycle of light and dark. So all these factors, because affect the epigenetic background, tend to make these mice very, very different. And so I'm not completely surprised that what you see is a difference between mice. I could not really interpret that, but I'm not surprised.
Yeah, Gene, I think I'm not at all, I'm less expert in this than Luigi, to be sure. But thinking of the human side, some of the data that Luigi showed about the differences between cross-section and longitudinal changes, and looking at people who went through major traumas, like people who lived through the Depression, are different than 60-year-old at 60 than a 60-year-old that never experienced that, maybe different. And again, there's such a major life trauma that in mice, how do you boil that down to mice? I bet if you were to look, I bet it's published somewhere, as sort of the conditions of rearing, of sort of raising these animals, you'd see the kind of differences. And then if you looked at some of the epigenetic changes that were taking place, it would be a very interesting look at some of those issues and the process by which they happen.
Mm-hmm. Okay. Yes, and I imagine, like you mentioned, as we start to look at and move towards translational studies in humans, that will probably very likely see those factors impact our results as well. So
I think that working on the cytoskeleton, I know you guys are working on the cytoskeleton and on the tubulins, and I think that that's an area that is really exploding. I think that especially in the context of what people call the integrated stress response, it's clear that that is part of the integrated stress response. And so I think that we will understand a lot more in the future of how important is that, in moving the different parts of the cell from one place to another, not only organelle, but even molecules. So I think that that's a fascinating thing you're doing.
Thank you.
We're coming up on 5:30. Does anyone have any last quick question? It's very late for you, Dr. Ferrucci.
It's fine. The coffee is working.
Are you in Italy, Luigi? Are you in Fiesole or where?
I'm in Fiesole, yes.
Yes, yes.
I'm in a part of the house that you've never seen because we bought this second part a few years ago.